Let's give him a warm welcome up to the stage. Last week.
It's great to be here.
It's been, I mean,
We try to pick up today. Left off last meeting 2 years ago and build on that. And that is modeling, putting together a model of the Great Salt Lake ecosystem that now includes nitrogen cycling, which has been one of the original methods that we started to look at in the early years of the technical advising group. Let me point out what I'm going to talk about today really builds on the Great Salt Lake ecosystem. Much of what I'm going to present has a lot to do with work of other people.
Okay, so I just wasn't sure. I'll go ask the gals at the desk.
So this is the updated food web or ecosystem model that we have for the lake. And last time at the Friends meeting, I focused on the pelagic, the open water part of this model, which is the phytoplankton, the shrimp, and the greens. Since then, the model has expanded to include the benefit of the microbialites, the brine flies, and the other birds in the system. And this is all complete now, except for the other birds in the system, such as phalaropes, which we have to now deal with. And we don't have the data yet to make all those connections in the model. This model is very, very large. 278 equations, 334 variables, and 17 constants. And those constants are more than one number because if you think of it, salinity is 12 months out of the year, so you got 12 values. Temperature is 12 months out of the year, you have 12 different values going into the model. Give you an idea of what the model looks like. All of these pictures that are here constitute a subroutine within the computer model. And this is the subroutine here for the phytoplankton in the pelagic section. Here is the growth of the phytoplankton. Green is the chlorophytes, the green algae. Blue is the cyanophytes, the cyanobacteria. And the brown here is the diatoms. Then this section that goes down here is the consumption of each of those phytoplankton classes by the brine shrimp, broken down to age category or developmental stage of the brine shrimp in the population. Here's a graph showing how things change for each of the types of phytoplankton. And since now we have the benthic portion, the microbialites built into the model, there is a linkage between the microbialites and the pelagic population, the phytoplankton. And that comes about as the brine fly larvae graze on the biofilm that make up the microbialite, and they're releasing algae into the water column. And that's what this lower graph is. This is the release of the algae that's in the biofilm of the microbialite up into the water column. Now, how does this model get its equations and its parameters? It requires experiments. The problem is if we went to the field, we have many field measurements from the Great Salt Lake Ecosystem Project. If we use the field data, things change together. So warm temperatures during the summer months lead to an increase in salinity in the lake due to evaporation. So you're not measuring things independently of what is the effect of temperature, what is the effect of salinity, what is the effect of nutrients on the growth of the phytoplankton and on the microbialite biofilm, what is the effect of temperature and salinity and abundance of food and type of food for the growth and survival and reproduction of the shrimp and of the brine fly larvae. So this requires experiments and requires many experiments. There are well over 10,000 laboratory experiments to get these independent measurements. And to give you an idea, that would be many pounds lighter and without gray hair. It has taken some time to get to the stage where we have these measurements. But anyway, to go on, the bird data has been put together by a number of people working with Mike Conover, part of the GSLEP program. And they measured consumption and consumption primarily, and other factors of the birds, grebes completed, and other types of birds that the phalaropes look to be added. But anyway, lots of data, the numbers are independent of each other. And this is what the model looks like and its output now. So let's look at the different parts of the model, and in particular the benthic part right now. And you can see that this is the predictions of the model in red. Blue is the observed values for the blue is observed, red is predicted, and what we get is, this is the biofilm chlorophyll, and this is in centimeters squared, because this is the surface. In centimeters squared, the amount of chlorophyll that's predicted for certain months of the year. We only have March, May, June, August, and December measurements from 2015 to 2023. And the model predicts it pretty well. This is for larval numbers on the microbial. And the red again is predicted, blue is observed, and that's predicted very well. And this would be the pupil numbers on microbial life, and that's predicted well. Linking the benthic with the pelagic, we find that the phytoplankton in the pelagic environment increases by 2.5% due to this grazing and release of the algae by the brine fly larvae. This increases the abundance of shrimp by about 2%, but it increases the production of cysts by about 4.8%. So it has an important aspect, influence on the reproduction. Adding the birds, the grebes in this case, we find that the grebes are reducing the shrimp population by less than 12%. And in fact, that reduction in shrimp numbers actually further helps to increase cyst production, because there's now less competition for food among the adults that are still remaining. And so the adults that are still alive, not being eaten by the grebes, are actually able to reproduce at a higher level. Finally, as I talked about last time to the Friends meeting, this is the predictions for the pelagic phytoplankton. Again, red is predicted, blue is observed. Phytoplankton production is predicted very well by the model. This is the nuclei numbers for each month of the year, juvenile numbers of shrimp for each month of the year. We missed this spike here, and adult numbers over time during the year. So the model does pretty well. Matter of fact, I don't want to say very well, but it's good. Now let's take that same model and use it to look at nitrogen cycling within the lake. The model as it's built right now assumes that the limiting nutrient for the production of phytoplankton and production of the biofilm on the microbialites is nitrogen. This is old-time 1840s observation of Liebig's law of the minimum. You know, a German agricultural scientist who found there were certain substances, one substance that always limited the growth of this agricultural crop. And if we look in aquatic systems, this is carried over to the idea of the Redfield ratio, which is the nitrogen to phosphorus ratio. And if that ratio is less than 16, nitrogen is the limiting resource to the production in the aquatic system rather than phosphorus. And given the number of measurements we have of nitrogen and phosphorus, inorganic nitrogen and phosphorus in the lake, we get an average value of about 4.6, indicating nitrogen limitation. Added to that, we have numerous experiments that we've done and experiments that others have done with Great Salt Lake phytoplankton showing that nitrogen is the limiting resource. And now adding that to the experiment or to the model, it blows up even larger to 408 equations, 517 variables, 33 constants. What data do we need to be able to measure nitrogen cycling? Well, we need a number of very different things than what we needed to get at the food web model. First of all, we need to get nitrogen content of the different pelagic environment components and of the benthic components. So in other words, we need to know how much nitrogen is there in milligrams per microgram of chlorophyll that you measure in the water. You need to know how much nitrogen exists per pellet. And since I'm not a storage engineer, I'm not going to use the technical term. We use poop. So we need to know how much nitrogen per poop. We need to know how much nitrogen per body of a shrimp, nitrogen per cyst, and then various values for the birds. And then over on the benthic side, we need the values for microbialite biofilms. We need the frass, that's the technical term we biologists use for insect poop. The frass nitrogen content, larval body content, pupal body content, and the salt body content. Then we need to know the amount that's produced. Well, a good part of the amount that's produced comes from the trophic dynamic model. What is the mortality? How many dead ones do you get per time period? And what is their growth? So they're adding weight. And what is their reproduction? But then we need another variable, and that's the fall rate. Well, first of all, let me point out, we got poop production here by the shrimp, and that's approximately 400 to 700 poops per week, per individual. For the brine fly larvae, poop is 739 poops per week. Now, we don't have to worry about poops by pupae and adults because they're not pooping in the water. Okay, but then the next thing we know is how fast does the product fall through the water? Well, the phytoplankton, when it dies, it essentially stays in the water column and will decompose before it settles out. However, brine shrimp poop falls 86.5 meters per day. They're little bricks. Falls and settles on the bottom in less than a day. Unbelievable. I would have never guessed it. I would have thought it would have stayed in suspension for a while. But we had to measure this in the lab over and over again because I didn't believe it. It was falling there. And then the bodies of the shrimp are falling at about 0.45 meters per day. Now, oftentimes they're up in with the cysts in the streaks, but those are caught in the surface film. If you're not in the surface film, you're falling pretty fast. And then we got fall rates for bird pre-poop, etc. Then over here, we don't need to worry about fall rates for the benthic community because it's already on the bottom. However, the biofilm, when it's grazed by the brine fly larvae, some of it gets put back up into suspension in the water column because it's broken off from the film that's covering the structure. So we had to understand that a good portion of that, rough guesses from experiments in the lab, is about 70% of what is grazed by the brine fly larvae gets back up into the water column. And then we have these terrible experiments that have to be done, which is decomposition. How fast does the particular product break down at different temperatures and different salinities? So this is the rate at which it breaks down. We've got this all completed now for the pelagic environment. We've got the bodies of the brine fly larvae worked out, and we've got preliminary estimates for the other components of the benthic environment worked out, but we don't have the detailed relationships yet with temperature and salinity. So then we got to rebuild the model and add to it what is released from the pelagic side. And when we look at the pelagic side, there are two types of releases. Releases that go into the water column, and releases that end up in the deep brine layer. And this is for the period of 2015 to 2020. So for each month, the model will predict how much is released by the pelagic components of the ecosystem into the water column. Then we have how much is added, the black line here, on a monthly basis to the deep brine layer during that period, and how much is then released from the deep brine layer back up into the water column. And that's what this relationship is, showing the turnover of that deep brine layer based upon the salinity of the pelagic level above the deep brine layer and what the deep brine layer salinity is. As salinity gets greater in the lake, there's more turnover because the deep brine layer and the water above it are similar in density. Then we can make a pelagic side of the model and look at how much is released each month. And this is in terms of milligrams of nitrogen per centimeter squared, not in terms of liters. This is on a surface. In both of the pelagic and the benthic portions of the model, we have a depth of the phytoplankton and biofilm of the microbialite. We have the release of poop and dead bodies by the shrimp and the larvae of the brine flies. And then on this side, we have how much of that is being picked up and put into growth for each of those categories and reproduction. And then we can calculate a net on the complete ecosystem. So how does this work? Well, first of all, here's our microbialite down here. Got our fly population. Here we have our biofilm. In pelagic, we have phytoplankton, and we have our shrimp. We have the poop here for the shrimp, and we have the poop here for the brine fly larvae. What goes on? Well, first of all, a lot of the dead biofilm that's released by the grazing brine fly larvae ends up in the water column. Certain proportion still stays down here on the surface. Because the bodies of the shrimp and of their poop fall so quickly, it goes down into the benthic environment. but a certain portion of that falls even further down into the deep brine layer. The model then tells us up in the pelagic, we have a flux of phytoplankton providing 0.29 micrograms of nitrogen per liter. The biofilm that's come up is providing 0.8 micrograms per liter. And that now is picked up and used by the phytoplankton for growth, reproduction, 0.74 micrograms, and 0.44 for shrimp growth and reproduction. Add that column up, the flux column up, got 1.15, and we have 1.18 for the pool. They should be equal. They come pretty close. We know from the highest measurements of nitrogen in the water column that the base nitrogen level should be, that's available, should be about 0.95 milligrams of nitrogen per liter. And that means that the turnover is a multiplier effect of about 1.23. That means that because things are cycling so quickly, it's equivalent to 1.23 times what that base nitrogen is. This is the economics term. You spend $1, and over time that buck magnifies as it goes through the economy and spent over and over again. That's what we're talking about here. Here's what we get on the microbialite. And we got the shrimp bodies and food quality, biofilm that's there, fly dead, fly components of the group. They're adding up to 4.84 micrograms of nitrogen per centimeter squared. And on the biofilm uptake, we got 4.91 and 0.04 for the flies. And it adds up to 4.84 versus 4.95. A little bit further off than in the pelagic, but still nearly balancing. And the multiplier effect here is 1.39 with a base of 3.5 if you convert this value here on a centimeter squared basis in the benthic environment. And that's the relationship between benthic conversion versus volume of water above, which you have to take into account on a centimeter squared basis. Now, we go to the deep brine layer. About 0.17 milligrams of nitrogen per liter falls into the deep brine layer. This is 2015 to 2020. And we estimate only about 0.01 milligrams of nitrogen per liter is released back into the water column above it. Now to the telling part of the story. The red line here is what is predicted on a monthly level of what you should measure in the water in the Great Salt Lake in terms of nitrogen milligrams per liter. The blue is what was measured as part of the GSLEP program by the U.S. Geological Survey. It matches up very, very well. The model seems to be indicating the important aspects of the nitrogen cycling going on within the lake. We look at, brown here is the release of nitrogen, the portion of nitrogen accounted for at any month. Accounted for in any month by the benthic. The green is the pelagic nitrogen. What's that telling you is that the pelagic system is producing most of the nitrogen in the spring and the summer. The fall and the winter, you're getting most of your nitrogen in the lake from the benthos, particularly the microbialites. Overall, within the year, 46.4% of the nitrogen comes from the pelagic, and 46.4% comes from the benthic. I don't believe they're exactly equal, but that's what the calculations give at this stage of the game. About 7.2% is coming from release again from the deep brine layer into the pelagic. Finally, the model predicts that about 17% of the nitrogen that's released in the system settles out into the deep brine layer per year from 2015 to 2020. And the measurement that we get is that the deep brine layer during that period was adding about 23%. So something is going on that leads to a little more of the nitrogen going into the deep brine layer than what we're predicting. And my guess is there's some lateral flow that's not being taken into account. Everything that I'm using here in the model assumes it goes straight down to the bottom. And if there's lateral flow, it can go over the edge and down into the deep brine layer. I think that's what makes up that difference. And don't ask me how to measure lateral flow in the laboratory to get at that number. I'm at a loss to figure that one out. But anyway, that's where we're at. But what this is telling us is that the benthic, the microbialites in particular, that part of the lake is just as important as the open water in terms of the cycling of nitrogen within the system. Now that we have a model like this, we have the ability to make a prediction, and I didn't do this because this is all new, but you could predict what happens if the microbialites all die off. What's that going to do to your nitrogen cycling within the lake? You have a model to be able to make a prediction. We don't want to see the microbialites die off, but this would tell us what we might expect to see if that happens. And that's the beauty of having this model in the same way that we have the food web model, and we could then predict what happens if the salinity goes to a certain level to the shrimp population and to the brine fly population. Now we can go one step further and say what happens to the cycling of a nutrient in the system. Somebody might ask, what about phosphorus? Well, adding phosphorus is no big deal. All you have to do is put in the constant now for the phosphorus content of all those variables. How much phosphorus is in the food? How much in the bodies? We have that. I just haven't built that in yet. The next big step, though, is for using this for management. And in that case, the model is being built into a game, where you can take it for an extended period of time into the future and change the variables, and then it'll tell you this happens the next year, this happens the next year, and over a 5-year period, I've limited it to 5 years, saying what's going to happen to the lake. And to believe it or not, it's harder to do the game, making the game out of this, than it is to actually model the equations. Because I'm not a game maker. But anyway, thank you. That's what I wanted to report. This is where the GSLEP program has proceeded to. And like I said, this is not just my work. It's a whole bunch of people that have been working on this for years, that we're just synthesizing and bringing this all together to make projections for the lake. Thank you.
Good morning. First of all, I'd like to thank Lynn for inviting me and the Brine Cooperative to facilitate my participation. I have been following since the 1980s what's happening here with Artemia in the Great Salt Lake. I've always been very impressed with the work that was done by the Technical Advisory Board, bringing all these different stakeholders together But I must say what I have heard yesterday and today, I'm very impressed. And really congratulations to the way you were tackling this, not only all the stakeholders, but you really do it in a holistic perspective. And this is where I'm very much convinced you were on the right path. This is an example for the world, as you will hear also in my presentation. So I'm supposed to talk about Artemia and the use in aquaculture. But I start with a slide of a shrimp cocktail. You probably wonder what has this to do with the Great Salt Lake and Artemia. Because you all remember the movie Forrest Gump, and you see the shrimp are coming from the Gulf of Mexico. But some of you in the room will remember that Forrest Gump, that was 30 years ago. And today, the shrimp that you were eating are not coming from the Gulf of Mexico, but they come from farms in Ecuador, in Indonesia, in Vietnam, in India, in quite a number of places. I got some figures from Thomas that it might be close to 90% of the shrimp that you are consuming in the United States is not coming from the Gulf of Mexico, but is coming from farms. And how does that come? And you see here also on the time scale, until say about 30, 40 years ago, all the seafood that we were consuming came from fisheries. But we have seen, so around end of the 80s, 90s, stagnation due to a lot of factors, overfishing, pollution in some places, etc. And we know that the demand for seafood didn't stop at that level in the 1990s. Population increase, higher consumption of seafood all over the world. And it's a very, very nice, I will not call it a coincidence, but it is very good that around the same time of the stagnation, we have seen that modern aquaculture, because some of you might think, oh, if I go back, maybe just in the 19th century, that's the beginning of aquaculture. You will see in the next slide, aquaculture has a very, very long history. But it is exactly at the time that we have seen the stagnation, that aquaculture has come up, and I think many of you realize that today, the seafood we consume, more than 50% is not coming from fisheries, but coming from aquaculture. No time to go into the details, but you just see here the evolution of the different groups that are farmed from the 90s up to 2020, the latest data from FAO, and you see a constant increase that already has gone on for quite a number of decades. I told you that aquaculture is nothing new. Can you imagine the first written handbook on aquaculture, 200 BC from Lee, and that was the integrated farming of rice paddies with carp. And you see also the definition, because people sometimes wonder, yes, what is fisheries, what is aquaculture? If you think, for example, about the mussel production in the Netherlands, they are just collecting the small mussels and putting them out in another area where there is more food. Well, this is considered aquaculture, as you see, intervention of man in the production of aquatic products. And you will not find in the textbooks, but I like to classify two types of aquaculture, what I call food aquaculture, and as you will see in a moment, business aquaculture. Food aquaculture, very, very long tradition, hundreds, millennia in fact. And that, why do I use the word food? This is production of food for local consumption. And that goes back to China, Asia, a number of countries in the world. Business aquaculture, and you hear the word business, so this is not to produce a product for local consumption. It might be for local consumption, but it's mainly as an income. Think about the catfish farming in the southern states, think about the salmon farming in Norway. The younger people will disagree with me that I still call this a recent development, but if you look at the time scale of aquaculture, so the 1960s, and the pioneers were the Japanese, and then quickly followed by the Western world, where today, in fact, we could say that there is more R&D activity in aquaculture in the Western world than in Japan, who was the pioneer. And what, now you might wonder, why did they wait until the 1960s to develop that business aquaculture? Well, it all had to do with limited knowledge on the biology. For example, how to reproduce shrimp or crab. You might think, put a male and a female together in an aquarium, give them good food. Many species, they need special behavioral. It needs to be a tank of a minimum diameter for the mating behavior, things like that. So biology, technology, of course, if you think about when you intensify the production, the problem with oxygen, so you need better equipment. And of course, crucial, and that links with the word business aquaculture, profitability. The Norwegians, who have been the pioneers and very successful with salmon, they choose salmon and not herring. They would not have a big profit in farming herring, but did it with the salmon. So just one or two slides, cage culture systems, freshwater, brackish water, marine, sophisticated ones. You would be impressed to see the new cages that they are using in Norway, that a storm comes up and the cages go 10 meters deeper where they are not affected by the waves and back to the surface when the conditions are favorable. And then the pond culture systems, as the slides that I showed you initially for shrimp, but also marine fish, freshwater fish, brackish water fish, quite important. But in that business aquaculture, remember when I was mentioning the word biology, still today, even after all these years of successful developments, the key, still today, the key to successful aquaculture is the controlled, profitable, high-quality production of the babies. The babies that you will put in the ponds, that you will put in the cages. When I say babies, these are not the newborn babies. These are the ones that, in the case of fish, it might be 2 months. In the case of shrimp, it might be just 1 month, that they are produced in what we call a hatchery, conditions where they are pampered, get the right food, the good conditions. And this has grown over the years. Remember what I told you, 1960s, 1970s, pioneered in Japan. And then we have seen the big commercial developments in the 1980s. Different parts in the world, marine fish, shrimp. Today it's a multi-billion US dollar industry. I'm talking just the hatchery section. I'm not talking about the end product. The end product, as you see, 10 million metric tons of product, fish and crustaceans. And finally, here you see the word Artemia, because this is where Artemia is playing a very crucial role as a starter food. Okay, some of the people in the room will say, well, for some species it's not the first food, but for all aquatic species grown in aquaculture ponds and cages, in the early stages, there is a period, be it 1 week, be it several weeks, where Artemia has to be offered as a unique substitute for the natural plankton. And why for the natural plankton? Well, this is the particular characteristics of Artemia, that they are producing these encysted embryos. We often call them eggs, but in fact it's a 4,000 cell embryo that goes, not the word dormancy is even not correct, they go in a state of, now it will be very contradictory what I'm saying, they go in a state of reversible death, because metabolism is zero. You store them for 50 years, experimental evidence is there. After 50 years, you put them in seawater, the next morning you have your plankton. And of course, that has been the reason of the very, very successful development of hatchery aquaculture, because you didn't have to rely, as the Japanese initially did, they were growing copepods, but that you could, I call it magic powder, that you can start from these dried cysts, and the next morning you can have as much numbers of plankton that you need to feed either your marine fish, freshwater fish, crab, shrimp, etc. So already in the 1960s, even before that business aquaculture started, there were two sources here in the Great Salt Lake. We heard from earlier speakers, Paul sitting in front of me here, referring to the Sanders, the Sanders Company, pioneers, and then very shortly thereafter also the San Francisco Bay brand in the San Francisco area. And that was at a time, believe it or not, where the world consumption was maybe maximum 100 kilograms of cysts per year. Why? Because it was only used in ornamental fish shops, people who had an aquaria with be it guppies or be it even seahorses, feeding them with Artemia. Business aquaculture, as you have seen that graph, going up to now over 100 million tons per year in total, but I'm referring to the 10 million tons where Artemia is being used. This has really expanded exponentially over the years, and so today we are at over 3,000 metric tons of Artemia cysts consumed all over the world in these hatcheries for shrimp, for crab, for marine fish, etc. 40%, there have been periods of more than 50%, 40%, but still today, and for quite a number of years now, nearly half of the world provision of Artemia cysts comes from the Great Salt Lake. There are a number of other areas, different salt lakes in Central Asia, Siberia, Kazakhstan, Uzbekistan, coastal salt works, and also inland lakes in China, and small, at this moment it is talking maybe 20-30 metric tons per year, so that nearly insignificant, but it has a very unique value and maybe some opportunities in the future, is in a lot of tropical, subtropical countries integrated in the salt production. But there are critical needs, and this is where I must say just in the last 2-3 years, and particularly in the period of COVID, that FAO, the World United Nations Food Organization, really reflected on where do we stand with Artemia, and they were, in fact, they were challenging me. Tell us, what about 20 and 30 years from now? This is such an important source of food production. Where are the guarantees that we will have the Artemia? And you see the critical needs, sustainable harvesting, and particularly when we are talking, and as you will see in the next slides, terminal lakes, protection of the Artemia habitats, preservation of the species. I'll just show you, and there could be more, but I'll just show you 2 or 3 examples. In Turkmenistan, huge lake, over 1,000 tons in 1995. You go there today, no more Artemia. Aralsea, 2,000, several thousand tons per year. 2019 already, eastern part dry, still a little bit of Artemia coming out of the west side. Urmia Lake in Iran, same story. So in other words, one has to be very, very concerned about the future, and the great thing there is that Great Salt Lake, Utah is an example, as I was mentioning in the beginning. For a number of years, in fact for a number of decades, you have strict protocols for cyst harvesting and are very consistent, as we have heard in the previous presentations. So FAO in 2021 came up with a reflection on sustainability issues related to Artemia. An international aquaculture consortium was set up, guided by FAO, coming up with a number of recommendations. First recommendation, particularly on sustainability issues. The consortium has played a role there. And September of this year, you see FAO is organizing for the first time a specific meeting, training on the sustainability issues for Artemia management, and I'm proud to show you that the work done here at the Great Salt Lake is used as an example. Recommendation 2, one has to look into certification, very crucial that we know where the product is coming from. Good manuals and material for the utilization, because many places in southern countries in the tropics are not doing it the proper way. And as the last slide, because my time is over, you might be surprised that in quite a number of countries, Bangladesh, Vietnam, Artemia biomass is being used now for human consumption. Artemia kebab is a new product that helps the malnourished children in Bangladesh for good feeding. And my time is over, but you can just read what is on the screen there. You should realize also that your brine shrimp Artemia here is playing a very important role in research. And I like this particular title of a colleague from China, From Ecology to Oncology. To understand cancer stem cell dormancy, ask a brine shrimp Artemia. Because they managed to, we did the genome sequencing of Artemia. The colleague in China has found the gene that is responsible for switching on and off life, has transferred it in breast cancer cell lines, and you can switch on and off cancer. Thank you very much.
Try to speak right into the microphone. Unfortunately for all of you, I'm not a biologist. I'm actually an engineer, an industrial engineer at that. So I can pretty much guarantee you that this presentation is going to be pretty boring. But anyway, I'll try and move through it pretty quickly and save you some time. What we wanted to do today is talk about the management of our resource, but more how it relates to our industry. So give you a bit of an industry perspective. I'll give you maybe a little bit of information that most of that was really positive for our industry. There are a few problems I want to highlight as well, but definitely that management has been overall really positive for our industry. So what do we want to show you today? What do I want to show you is maybe just revisit very briefly the importance of Artemia and Great Salt Lake Artemia in world aquaculture, and then talk about the resource management, but how it relates to our industry specifically, and then more specifically talk about the harvest management as well as the salinity management, and then maybe end with some closing remarks. Okay, so let me dive in. Why is Artemia and Great Salt Lake Artemia so important to the aquaculture industry? As Patrick was explaining before me, total aquaculture in the world today is approximately 110 million metric tons of food production a year. Now, a significant portion of that is algae and mollusks, excuse my Belgish here, but a significant portion is also fish and crustaceans, mostly shrimp. 64 million metric tons of fish and crustaceans are being produced a year in the world. Of that, about 19 million metric tons are marine species, and of that, more than 10 million metric tons of food relies on Artemia for its production, especially in that early production cycle, as Patrick was mentioning. Now, what does that mean for the Great Salt Lake? This is a graph that shows you the production, the world production of Artemia. In yellow, the Great Salt Lake. In blue, the Central Asian countries and Siberia, and in gray, China. And you can see that the world production, at least in the last decade or so is approximately 3,200 metric tons of Artemia cysts, dry Artemia cysts produced a year. Our Great Salt Lake produces about 1,500 metric tons of that, so close to half. So putting that back in relation to aquaculture, if you have 10 million metric tons of aquaculture, marine aquaculture, that relies on Artemia, and half of that comes from the Great Salt Lake, you'll have more than 5 million metric tons of marine aquaculture relying on Artemia from the Great Salt Lake to be produced. What does that really mean for you? Because with shrimp, for example, it's actually nearly 100% of the shrimp that rely on Artemia to be produced. Well, if you go to a restaurant and you order shrimp, or if you buy shrimp in a supermarket, you have to think of it this way: there's about a 50% probability that that shrimp has eaten brine shrimp from your own Great Salt Lake in its very early life cycle. So it's actually really quite significant, the importance of our Great Salt Lake when it comes to worldwide aquaculture. Okay, so segueing into resource management. First thing I want to talk about, very briefly, just point out what are the factors that affect our brine shrimp population, factors that we have some control over. That is harvest management, salinity management, and to a certain extent, even nutrient management. Today, I just want to talk about this harvest management and salinity management. A lot of research has been done on our ecosystem and how the harvest management actually influences the Artemia population, but also the species that rely on Artemia. And ultimately, that management is based on a unimodal curve, and what it boils down to is that we're trying to reach what we call an optimal escapement stock, 21 cysts per liter. That is sort of the magic number that we're trying to reach at the end of the harvest season. And that assures us that we get the best possible population the subsequent year, and the best possible also cyst population in the subsequent year. So what has this done for our harvest? Because the whole research has already been focused on what does it do for the ecosystem, for the brine shrimp population, the avian population that relies on the brine shrimp and so on, but what does it do for our industry? This graph gives you an idea of the production of Artemia over the years, and in that early period when we first implemented that sustainable harvest system, sort of the late 1990s, early 2000s, we still saw quite a lot of variability in the harvest. So this graph, it shows you the year-over-year harvest, and I have to say it's a dry equivalent harvest. Just keep in mind that the dry harvest is only about 10% of the raw harvest that we report to the Division of Wildlife Resources, because there's a lot of water that we harvest, a lot of non-cyst material that we harvest. But you see that variability in that first period of time. And then, as we get better at reaching that optimal escapement stock of 21 cysts per liter, you start to see more stability in the harvest. And if we focus a little bit on the sort of the variability over time, you can see that because the standard deviation over time from a harvest to harvest is actually going down, indicating that we really are getting a more reliable and a more stable harvest. If you look at the regression of the harvest year over year, you see sort of a slight increasing trend, but it's not significant. But again, it's an indication that overall the average harvest is really quite stable over time. Now, in this period of time where we get more stability, we do see a little bit of variability. And the variability is primarily the result of events that we don't control, mostly weather events. I won't dwell on that too much, but there's a few examples here. In some cases, we're just not able to harvest the product because of weather. In some cases, the weather is great and we get an additional population of Artemia being produced, and therefore we can harvest more. So these are really events that we don't control. There are a couple of harvest years where we did experience a lot of problems. and we'll come back to that, where salinity, we believe salinity plays a role. But you would ask yourself, well, why didn't you just keep harvesting until you reach that optimal 21 cysts per liter escapement stock? We did, but the problem is we lose cysts to the system. Cysts that break dormancy actually start hatching, and so then we can't harvest them anymore. So if we don't get the opportunity to harvest them, then we don't. So you get a little bit of variability, but at the end of the day, overall, we get a more stable and a more reliable harvest. And that, for us as an industry, is really important. It's really important because it allows us to plan better. What kind of operations do we need to plan for a harvest? Because we can rely on a pretty stable harvest. It also allows us to plan our inventory management in a better way, and therefore our sales to the market. So, segueing into the salinity management and the salinity effect on Artemia, and that's the bad news, and I had to bring some bad news into this presentation, but rest assured there's some good news to follow as well. So here's the bad news. We do know from the literature that there's an optimal salinity range for Artemia. We know that at a higher salinity, above about 160 grams per liter, we get a decline in the population, real population level effects, and below about 90 grams per liter, and it varies, but we start to see effects of predation. Now the predation effect we saw drastically in the 1980s, where salinity went below 50 grams per liter, and the brine shrimp population was decimated by the corrosive at that time. We hadn't in recent years, at least in years where we've been able to collect a lot of data, we haven't been able to see what this higher salinity does specifically to the brine shrimp population on Great Salt Lake. So we had a lot of literature about it, but we hadn't experienced it ourselves, except for 21 and 2022. And what this graph shows you is in these orange and blue dots, it shows you the salinity over the last 8 years or so. And in that sort of blue circle, you can see that that salinity actually, spring salinity is increased above 130 grams per liter in those 2 years, and fall salinities were as high as close to 180 and 190 grams per liter. Clearly above these critical levels where it starts to affect the brine shrimp population. What did we see as a result of that? And I come back to those 2 years where we saw below average harvest. We did see a below average harvest. Probably a lot of factors that played a role there, but we do believe that at least one of the factors that played a role is the onset of the population. So the whole population was delayed during that period of time. And we can see that spring hatch being delayed by approximately 2 months. If you look at that graph to the left, the white line shows you the long-term average emergence of Artemia nuclei, and the yellow graph shows you the emergence of Artemia nuclei in 2022. And you can see that big delay in the onset of that population. On the graph to the right is very similar. It shows you the next cohort of adults following these Artemia nuclei. So we see a delay in the population that has potential repercussions for the biota that relies on the Artemia from the Great Salt Lake. One of the reasons we think that may have happened, again, they're all hypotheses, but this one is probably a high likelihood. In our studies, we can see that the Great Salt Lake Artemia actually doesn't hatch very efficiently anymore at higher salinities. Once you reach that salinity above 130 grams per liter, PPT in this case, you start to see a very, very reduced hatch, and by the time you reach 140, 150, there's virtually no hatchability. So that really slow hatchability, we think was one of the reasons why we saw that delay in the population. Is that an issue for our industry? Certainly it's an issue, because the average harvest goes down. But is it existential? No, it's not really existential. We can still deal with that. The problem that it created for us, what we can't deal with anymore, again during that same period of time, we saw a huge effect of that high salinity on the quality of that offspring, on the quality of the Artemia cysts. And here, this graph shows you again the harvest over time, but then in the red line, you'll see the average hatchability of the harvest over time. And typically, Artemia from the Great Salt Lake, average harvest hatches about 75%. And then you see those 2 seasons where we had that high salinity, and suddenly you see that hatchability go down to 59% that first year, and as the salinity increases even more, the hatchability goes down to 49%. And that is existential to our industry. Because you have to think about it, Great Salt Lake Artemia is known around the world as a high quality, top quality Artemia, and an Artemia that's really easy to manipulate. It's really easy to hatch. And then suddenly we're losing that high quality. That one most important factor in our sales, we're losing that. That means that we can't compete anymore with these cheaper sources from around the world. And that is existential to us. And so, what did we learn? We learned that yes, that there is a delay in that spring hatch, there is a delay in the population. We also learned that there's decreased hatchability of the cysts that are being produced and of the overwintering cysts, and that's the existential problem for our industry. And frankly, had that continued for very much longer, we would have had to just dry dock our boats, and we would have had to mothball our operation. We just couldn't have continued like that. And it was only because we're really careful about inventory management that we were able to span this period of very high salinity. Okay, so now the good news, enough doom and gloom. Here is some good news, and that good news is, believe it or not, the adaptive management burn. So, the adaptive management burn was put in place in 2017. We know that leading up to 2017, we lost the connectivity between the north and the south arm as a result of the culverts failing. A breach was put in place to re-establish that connectivity, and thanks to the foresight of the Great Salt Lake Advisory Council, when that breach was constructed, the state also decided to put in place or to construct an adaptive management berm that allowed us to somewhat adjust the flows of brine between the north and the south arm as a tool to try and manage salinity. And going to the next slide, what this graph is showing you is based on our salinity measurements, we were able to at least get an estimate of the total salt mass in Gilbert Bay, in the south arm of the Great Salt Lake. And you can see after the opening of the breach, at first we see a huge reduction in that salt mass. And that's because there was a big head difference between north and south. We export a lot of water to the north arm, and with that water came a lot of salt, or a lot of salt was exported to the north. Then we have a period of time of more or less stability between export of salt to the south and re-import of heavy brine, export of salt to the north, and re-import of heavy brine to the south arm. And then as that head difference subsided, then we start to see a re-import of salt into the south arm from the north. So there's heavy brine actually flowing back to the south from the north arm. Now, as a result of that, if you combine that increase in that salt mass in the south arm and a decrease in lake volume, suddenly that is why we were reaching these salinity levels that were really critical and that had this population level effect on both the life cycle of the population, but also the quality of the Artemia cyst. Now, as a result of that increasing salinity in the summer of 2022 and subsequently in the spring of 2023, the state decided to adapt the management berm. First, to stop the return of that heavy brine coming to the south, to help mitigate that salinity problem. And then again, a second time to capture the spring inflow, create better mixing, decrease the salinity again, and then being able to export some of that salt to the north. And what do we see on our salt mass calculations is we see that the salt mass again starts to decline. And it starts to decline in at about 5% a year. So now, under those conditions, we did have a lot of spring runoff, but under those conditions, salt mass declined by about 5% a year. What does that mean? That means that if the lake had been at the maximum salt mass, at a level of 4190 feet, it would have taken about 4 years for that salinity of 190 ppt to come back down to that more acceptable level of 160 solid grams per liter. Now, what did we learn from all of that? Well, we did learn that we can effectively use that management verb to manage salinity, both bringing the salinity up and bringing the salinity back down. We also learned that it's a pretty slow tool. And we've been really thankful for the last couple of years of inflows and above average winters, because it's given us, at least as an industry, the opportunity to get a decent harvest again, and given us some time, or given the state some time to continue to work to decrease that salt mass in the south, so that if this drought continues, we can at least maintain the salinity levels within acceptable ranges for the Artemia population. So, with that, maybe some final thoughts to wrap it up, at least try and summarize. Sustainable harvest management definitely results in a more reliable and stable cyst harvest, which is very important to us as an industry. Secondly, specifically for our Great Salt Lake, salinity is above 160 grams per liter in the fall, at the high end, above 130 grams per liter in the spring when the hatch takes place. Those are salinities that become critical, not only for the length of the population cycle, but for us, really existential and critical is that it affects the quality of the offspring. Now, we did learn that the breach that was implemented as a management tool really does function well as a management tool. It's amazing, it works. And that's basically a savior for our industry. And then I do want to make a bit of an announcement, but these 3 decades of work that the whole community has done on Great Salt Lake has ultimately culminated in last year our Artemia fishery obtaining Marine Stewardship Council certification. And thanks to all of you. It's really quite amazing, and I'm really proud to say that it is the first Artemia fishery in the world that obtained that certification, and it's also the first inland fishery in the United States that has obtained that certification. And with that, I really want to thank all of you for these more than 3 decades now of dedication to both the research and the management, and for your support of our Great Salt Lake environment, our Great Salt Lake biotope. Thank you. And I'm happy to take some questions if there are any. 2 minutes for questions.
Did that late hatch, was there any temperature effect? I think of temperature as one of the main driving factors for the spring hatchery.
Yeah, definitely, temperature is definitely a factor, there's no doubt. And when we showed you that graph that shows the average, there's a little bit of variability there, Wayne. But this was a 2-month delay. And so what happens is, in order for these Artemia to be able to hatch at these really high salinities, they have to rely on precipitation. They have to rely on inflow. And that is a much less effective way of restarting that population. At least that's our hypothesis, Wayne. I mean, there's still a lot of research to be done to confirm all of that. But that's our hypothesis there.
I'm not sure. So if you control just the salinity, what does that mean to the volume of the lake? Let's say the lake keeps drying up. Does that mean the north arm will get completely dry and then control salinity, controlling the level of the lake?
Yeah, absolutely. So the salinity control, it's a stopgap measure for us to start getting more water into the lake. And there's been a lot of presentations in the past, yesterday, and I think there might be some more about, how we're going about trying to get more water into the lake. And then there is, of course, I think it was Representative Schultz, the Speaker of the House, that said that there is a minimum level at which the north arm would indeed be cut off from the south arm in order to capture and maintain all the inflows in the south.
Oh, thank you for the wonderful presentation. It was definitely not boring.
Thank you.
Aside from the burn management, are there any particular policy tools that you see as being helpful in assisting the continued sustainability and success of the Brine industry?
The harvest management for sure, the burn, the nutrients. I didn't have time, but there were some really interesting things that Division of Water Quality has done to maintain nutrient inflows into the lake. One of them is the pipeline that was built to move the discharge of one of the POTWs from one area of the lake to the hypersaline area of the lake where the nutrients are really positive. They don't negatively affect water quality. That was an amazing thing. Other things that have been happening at the level of DWQ, in determining nutrient criteria, the way that these criteria have been developed, also really quite unique in the United States. In order to try and not be too stringent, but be stringent enough to protect the beneficial uses, but at the same time allow some of these nutrients to come to the lake where they're really necessary.
Hi everyone. The mission of Friends of Great Salt Lake is to preserve and protect Great Salt Lake through education, research, advocacy, and the arts. And I do want to emphasize that art is very important at Great Salt Lake. Friends of Great Salt Lake has an excellent arts program, the Alfred Lambourne Arts Program, and you can get more information about that outside. And we'll have our program for that on September 6th. So I just want to give a shout out to Art and the Alfred Lambourne Arts Program before Sam and I talk about another of our great programs, the Doyle Stevens Scholarship Award Program. Just really briefly, I'm Molly Blakowski. I am a PhD candidate in watershed sciences at Utah State University, and I'm a physical scientist at the USGS Utah Water Science Center. I'll let Sam introduce himself as well.
Hi everybody, I'm Sam Lopez. I'm a PhD candidate at the University of Utah in the Geology Department, and I'm also a hydrologist at the USGS Water Science.
So it's a real honor for Sam and I to present this year's winners of the Doyle W. Stevens Scholarship Award, because we've both won this award ourselves, me in 2020 and Sam in 2022. There are a number of other past recipients in this room as well. Can you guys stand up? This is who you let cut to the front of that long lunch line, folks. There's also a number of regular donors in this room. Can we give them a round of applause as well? Another reason why Sam and I are really proud to make this announcement is that Doyle would have been our co-worker and no doubt our mentor. Our colleagues at the Utah Water Science Center who remember working with Doyle said he had his hand in just about anything related to the biology of Great Salt Lake and Salton Sea. Steve Gerner actually dropped a box full of Doyle Stevens reports and memos into my cubicle the other day, and I'd like to read an excerpt from a report he wrote about Salton Sea, I think back in 1999. If our answer is the survival of wild things, then we need to reject the position that human-altered ecosystems are of less value than more natural ecosystems. Allowing the Salton Sea to go seed because it is an artificial system will jeopardize the well-being of a number of avian species. Further, if one accepts that human-altered ecosystems should not be actively managed for wildlife, then in general, we will need to let wildlife fend for themselves, since so much of the habitat base for wildlife is either altered or artificial. Rejecting such habitat because it is artificially maintained is inconsistent with the conservation of wild things. There are many footprints in the Salton Sea Basin, but one of the greatest current significance belong to Homo sapiens. We have reached another crossroad regarding which species will survive and which will be banished from this environment because of the challenges associated with water availability. For migratory birds, the Salton Sea is part of a large landscape that has become increasingly impoverished relative to wetland habitat. The sea has become an increasingly important area for wild things because of these losses. Our challenge is to learn how to improve the quality of this environment for wild things and human alike, and to sustain this system as a healthy environment for the next century. Aside from Doyle's incredible science and advocacy, he loved gin and tonics. Multiple people told me this, so he must have really loved them. I was also told that while he wasn't the loudest voice in the room, he was the one who everyone gravitated to. He was a free thinker and loved by all. With that, I'll let Sam announce this year's winners.
Thanks, Molly. Just to add one thing about Doyle Stevens, when I first started at the Utah Water Science Center, I had a chance to flip through some of his old field notes. It was really interesting reading through his notes. He was thinking about a lot of the things with regard to Great Salt Lake that we're discussing today at this very issues forum and past issues forum. So, yeah, really nice to be here. Our first winner this year is Mei Chen from BYU, and her project is titled The Potential Unanticipated Lasting Effects of Glyphosate Control of Phragmites in Calcareous Wetland Ecosystems. May is currently traveling, but her advisor, Rachel Wood, is here to say a couple words on her behalf.
Thank you. I'm so pleased to be here. May really was disappointed. She is visiting family in Taiwan right now, but she is an awesome student. She graduated from BYU with an undergrad in environmental science and is a first-generation college student, and is currently doing a PhD with me in biology. And just a little bit about her project. So glyphosate is the most commonly used herbicide worldwide because it's highly effective. And it works by disrupting the shikimate pathway that is used by plants, bacteria, archaea, protists, in order to produce folates and essential amino acids. Recently, so while it's typically been shown that it degrades really quickly, like days to weeks or maybe months in soils. Some recent work by Lee Ferguson's lab from Duke University found that in arid calcareous soils and hard water that it could persist for 7 years in water and 22 years in calcareous soils. And so May, when she found that, she came to me and was as a soil scientist, like with her background in soils, that I think that we have some arid calcareous soils here. So maybe we could take a look just to see if this is something that's persisting. And because it also affects microorganisms, it potentially also has effects on microbial mediated processes. Like a paper published just this month has shown that it affects the rate of greenhouse gas emissions from wetlands. So this just highlights the need to use our tools as effectively as possible, because this is like the main tool that we're using to combat Phragmites, which is really important for us to control that invasive species. And so it's making sure that we're using our tools in the best way. And here we have land managers who are really committed to this, and Karin and Keith are going to talk more about that tomorrow. But anyway, that is just a little bit about what May is going to do. Thanks.
So our next recipient, I happen to know fairly well. I share an office with him at the University of Utah, actually. Our next recipient is Sam Carter from University of Utah for his project titled Using Environmental Tracers to Develop a Conceptual Framework of Groundwater Inflow into Farmington Bay, Great Salt Lake, Utah. If you were here yesterday, you likely saw Sam's presentation on this topic and some of his ongoing research, which is very high level, and a really nice contribution to current Great Salt Lake science. Sam's advisor, Kip Solomon, was unable to join us today, but he's shared a few words with us that I will read for you all. I was very pleased to learn that Sam was a recipient of the Doyle W. Stevens Research Scholarship. Having personally worked with Doyle many years ago, when I was an intern at the USGS, I am confident that Sam's research would interest him greatly. Sam is studying groundwater input to Great Salt Lake, particularly in the vicinity of Farmington Bay. Remarkably, groundwater contributions to Great Salt Lake are not well known, and existing estimates have large uncertainty. Sam aims to employ environmental tracers, trace substances and natural precipitation that can speak to groundwater ages and rates of movement in an integrated way that is not possible with traditional hydrologic measurements and water budgets. Already, Sam's work has shown that potentially 1 trillion gallons of fresh water is located beneath Great Salt Lake based on microconductivity measurements on pore water from cores collected beneath Farmington Bay. Sam is a dedicated and hardworking student. After completing a University of Utah undergraduate degree, he worked for the National Park Service and a private consulting firm, earning praise from each. He started working on his thesis this past summer before officially starting his graduate program at nights and on weekends since he was employable. He successfully interacted with state and federal agencies regarding existing well data and formulated an excellent working hypothesis regarding the interface between fresh and salt water in the vicinity of Great Salt Lake. He has continued to demonstrate sound scientific judgment, processing pore water samples, and details of calibrating instruments over a large dynamic range. Sam is an excellent choice for this important scholarship. My thanks and admiration to everyone involved in making this possible. Congratulations, Sam.
It's lunchtime now, right? Well, go enjoy.